Welcome back to the deep dive. Today, we are tackling a foundational paradox in cell biology. It's really the engine of evolution itself.
It really is. It's a fascinating puzzle.
Our mission today is to understand how life's most successful strategy, sexual reproduction, somehow manages two completely opposing yet equally critical tasks.
And what are those?
First, keeping the chromosome number perfectly constant across every single generation, and second, at the exact same time, generating this massive, vital genetic diversity.
Right. Stability and chaos, all in one process.
Exactly. If you've been with us for previous deep dives, you know that the division we call mitosis is, well, it's a high-volume copy machine. It's designed for proliferation, growth, repair.
It just churns out identical copies.
Right. Works great for a leaf cell or an amoeba.
Yeah.
But sexual reproduction demands something far more complex. It requires this precise programmed reduction followed by a dramatic reshuffling.
It really is the ultimate chromosomal balancing act. We have to address the fundamental arithmetic of life.
The arithmetic of life, I like that.
Every organism that reproduces sexually depends on having a consistent, protective set of chromosomes. This is what we call the diploid state, or 2N. So you, the listener, your cells are all 2N.
Meaning two complete copies of the genome.
Exactly. One inherited from each of your parents. And functionally, this diploid state it's like an evolutionary insurance policy.
An insurance policy? How so?
Well, think of it as having redundant backups for every piece of critical software on your computer. If you get a detrimental mutation in one copy of a gene, or if a chromosome suffers some kind of damage,
We have a backup.
you have a perfect high-fidelity template right there, the second homologous chromosome. It provides protection against gene failure, and it serves as the perfect scaffold for DNA repair. The stability is it's absolutely critical.
Okay, so if we accept that stability, that 2N state is paramount, then we immediately run into a huge problem when two parents want to combine their genetic material.
A huge problem.
If the parents are both diploid, 2N, and they just fuse their entire genomes together, the resulting offspring would be 4N.
And the generation after that, 8N.
It's completely unsustainable. Biologically, it just can't work.
Exactly. So you need a mechanism to have that chromosome count before fusion. And that mechanism is meiosis.
The star of our show today?
The star of the show. Meiosis takes a diploid parent cell and produces these specialized haploid cells, which we designate as N, or 1N.
Yeah.
These are single set cells.
And these are the gametes.
The gametes. In animals, that's sperm and eggs. These are the only cells in your body that are truly haploid. So meiosis is the process of going from second N down to N.
The reduction.
The reduction. And then when a haploid sperm fuses with a haploid egg during fertilization, the chromosome number is perfectly restored. N plus N gets you back to 2N.
Creating the diploid zygote.
Creating the zygote. So meiosis and fertilization are the twin pillars of sexual reproduction. They maintain the chromosome budget while at the same time providing all the raw material for adaptation.
Okay, let's unpack that strategic trade-off a little more because it really dictates why all the cellular machinery we're about to dive into is even necessary. On one side, you've got asexual reproduction. You said it before, it's the definition of efficiency.
It absolutely is.
Yeah.
It relies on straightforward mitotic division, so it just produces genetically identical offspring, clones.
Right, think of a bacterium just dividing or a plant you can regenerate from a cutting or simple budding.
It's fast, it's very low energy, and it ensures the immediate perpetuation of known successful traits. If it works, copy it.
But that efficiency, that's a trap in the long run, isn't
It's
it?
a huge trap. If the environment changes, let's say a new pathogen emerges, or the climate shifts dramatically, a clonal population that lacks variety might be completely unable to adapt.
They're all the same, so they're all equally vulnerable.
Exactly. They could face extinction very quickly. And this is where you see the massive evolutionary punch of sexual reproduction.
The variety.
The variety. By constantly combining and recombining genetic information from two dissimilar parents, the process generates offspring that are all uniquely distinct. This enormous variety gives the population options.
So even under really severe selective pressures,
There's a much higher probability that some individuals will have the right genetic toolkit to survive and flourish.
so all the complexity of meiosis, it's really the price organisms pay for that long-term adaptability.
It is. And as we said, that Ticonton balance is non-negotiable. But how that balance actually plays out in the overall life cycle is, it's remarkably diverse across the different kingdoms of life.
Right. It's not the same for a fungus as it is for us.
Not at all. The process of meiotic reduction and fertilization doubling is universal, but the prominence of the haploid versus the diploid phases can vary greatly.
So, for example,
Okay, let's start simple. Bacteria are exclusively haploid. They're always 1N. Any genetic sharing they do happens through specialized non-sexual mechanisms we can get into later.
okay, and what about fungi?
Fungi are fascinating. Their life cycle is often dominated by the haploid phase. They have a very brief diploid moment right after their gametes fuse, and then they almost immediately perform meiosis.
So they don't hang around in that 2N state for long.
Not at all. And we call this sporic meiosis because the result isn't gametes directly, but haploid spores. These spores then grow into the main body of the organism, the mushroom you see, for example, which is haploid. Then that haploid organism creates new gametes, but it does it by mitosis.
That's a huge difference. They make gametes with mitosis because they're already haploid.
Exactly. Now, if you look at plants like mosses and ferns, they have this amazing concept called alternation of generations.
Where both forms are prominent.
Right. You have both a haploid form, the gamophyte, which produces gametes, and a diploid form, the sporophyte, which produces spores. They are both distinct, visible organisms. Now, as you move up the evolutionary tree to flowering plants, the diploid sporophyte phase becomes much more dominant.
Yeah.
The flower you see is the sporophyte.
And then finally we get to us, animals.
And when we get to us, we are the classic example of a life cycle that is almost entirely dominated by the diploid phase. The haploid phase is confined exclusively to the mature gametes.
The sperm and the egg.
That's it. They are produced directly by the meiotic event. We call this gametic meiosis. So for us, where our haploid number N is 23 chromosomes, every single cell in our body is 2 N's, or 46 chromosomes, except for the sex cells.
Okay, so we know the why. Balance the chromosome count, create variety. Now let's get into the how. The machinery of meiosis is, fundamentally it's 2 divisions, but they're built on just one initial round of DNA replication.
That's the key. 1 replication, 2 divisions.
So a diploid cell starts in G1. It's 2N, and let's use the term 2C, where C is the amount of DNA in a single haploid set. After the S phase, the DNA replicates. So you have a cell that is still 2N, but it now has a DNA content of 4C.
Right. Because every chromosome is now duplicated, consisting of 2 sister chromatids joined together.
And the ultimate goal is to convert that single 2N 4C cell into 4 final haploid cells, and each of those needs to be 1N 1C.
To get that massive reduction, the process is split into 2 parts. First up is meiosis of Panda. We call this the reduction division.
Reduction division. Well,
Because this is where we separate the homologous chromosomes, the maternal partner, from the paternal partner. By separating the partners, we instantly have the chromosome number from second down to 1N.
okay, so the ploidy is cut in half.
But, and this is crucial, the individual chromosomes at this point still consist of 2 sister chromatids. So the DNA content only goes from 4C down to 2C.
I see. So you've reduced the chromosome number, but each one is still duplicated.
Precisely. And that's why we need meiosis II, which we call the separation division.
To separate those sister chromatids.
Exactly. The second division looks remarkably like a normal mitotic division, but it's happening in a cell that already has half the chromosome count. So by separating the sister chromatids, we achieve the final reduction in DNA content from 2C down to 1C.
This is such a key insight. All the unique, dramatic, world-changing events that define sexual reproduction, the pairing of homologs, the physical exchange of genetic material, the reduction of ploidy, all of it.
It all happens exclusively during meiosis I.
Right. Meiosis II is basically just the cleanup crew.
In a way, yes. It's the final separation. But the real magic, the part that generates all the novelty, is in meiosis I.
So let's zoom in on that. The action begins immediately in prophase I, and this stage is famously long and complicated.
It is. And it's long for a very specific reason. The cell has to perform its most elaborate feat of self-assembly. It has to get the 2 homologous chromosomes to recognize each other and then pair up exactly along their entire length. This process is called synapsis.
And that pairing forms a unit.
A unit called a bivalent. You might also hear it called a tetrad, which just refers to the 4 total chromatids that are now involved, 2 on each homologous chromosome.
Now, traditionally, this stage is broken down into 5 phases, right? Leptotene, zygotene.
It is, and the names are based on what scientists first saw under a light microscope. But I think it's more insightful to think about it functionally. What is the cell actually trying to do?
Okay, so functionally, what's step 1?
Step 1 is establishing contact. This covers the first couple of stages, leptotene and zygotene. The DNA condenses into long threads, and the homologous partners actively seek each other out and begin the pairing process, forming that bivalent structure.
And this initial pairing isn't just a loose association. It relies on some pretty incredible molecular scaffolding, doesn't it?
Absolutely. The main structural component here is called the synaptonemal complex, or SC.
Mm.
And you shouldn't think of this as a loose tie. You should think of it as a massive, elaborate molecular zipper, or a vice grip. It's this protein structure that literally locks the homologous chromosomes together, side by side, ensuring they are perfectly aligned across their entire length.
How do they even find each other in the first place inside the nucleus?
That's another great piece of structural biology. The ends of the chromosomes, the telomeres, all cluster together and attach to the nuclear envelope in a configuration that looks like a bouquet of flowers. This clustering brings the ends into close proximity, which greatly facilitates the initial searching for the right partner.
So once that pairing is locked down by this protein vice grip, this synaptonemal complex, the stage is set for the most critical genetic event. And that happens in the Paxton stage.
Precisely. This is the stage of crossing over, also known as homologous recombination. While those homologs are held so tightly together by the SC, non-sister chromatids, so one from the maternal chromosome and one from the paternal, physically break and exchange large segments of DNA.
This is literally where the maternal and paternal genomes mix and match.
This is the moment it happens. After that exchange occurs during Paxton, the SC begins to disassemble. The vice grip releases.
Okay.
And as the homologous chromosomes start to separate slightly, in the next stage called diploid, they remain physically attached, but only at the specific points where crossing over occurred.
And those attachment points are visible.
They are. Under a microscope, they look like X-shaped structures, and they're called chiasmata. If there's only one, it's a chiasma.
And these chiasmata are the physical proof that an exchange happened.
They are the physical manifestation of a prior crossing over event. And this is where we see that fascinating biological timing you mentioned earlier.
The pausing.
The pausing. In female mammals, the entire meiotic process can literally freeze right here in the diploid stage.
For how long?
Sometimes for decades. In human oocytes, it can be 40, 50 years. That cell just sits there until ovulation or fertilization signals it to resume.
Which means those chiasmata, those physical connections, have to hold those chromosomes together for all those years.
For years, waiting for the final signal. It's incredible. Finally, in the last stage, diakinesis, the chromosomes reach their maximum compaction, the chiasmata are the last physical links holding them together, and the cell prepared for division by dissolving the nuclear envelope and forming the spindle.
Okay, so that's the end of a very long prophase I. Now we move into metaphase I, and this is where the second major engine of genetic diversity kicks in, random assortment. In metaphase I, the bivalents, those paired homologous chromosomes, held together by their chiasmata, they all line up at the center of the cell, the spindle equator.
But, and this is a crucial distinction from mitosis, they don't line up single file.
They line up in pairs.
They line up in pairs. And the key mechanical difference that ensures this is the orientation of the kinetochores.
The little protein handles the spindle fibers grab onto.
Exactly. In mitosis, the kinetochores of sister chromatids face opposite poles. But here in meiosis the kinetochores of sister chromatids face the same pole. It's the kinetochores of the homologous chromosomes that face opposite poles.
Ah, so that structure guarantees that the homologous partners will be pulled apart from each other.
But the sister chromatids will stay glued together and travel as a single unit.
And the random assortment part comes from the fact that the orientation of any single one of those pairs is completely random.
Completely random and independent of all the others.
Whether the maternal chromosome 1 is pointing north or south has absolutely no bearing on whether the maternal chromosome 15 is pointing north or south.
None whatsoever. And you have to think about the profound consequence of this randomness. In us humans, we have 23 pairs of chromosomes.
So the number of possible combinations is 2 to the power of 23.
2 to the 23rd power, which is more than 8 million.
Wow. Over 8 million different uniquely assorted combinations of maternal and paternal chromosomes just from this one step.
Just from random orientation at metaphase I. Yeah. And that's not even counting the added layer of variety you get from all the crossing over that just happened. The potential for diversity is just astronomical.
Okay, so then comes anaphase I.
In anaphase I, the connections at the chiasmata are resolved, and the homologous chromosomes finally separate and are pulled to opposite poles. This completes the reduction division. The cell is now formally haploid, or 1N.
Even though each chromosome still consists of 2 sister chromatids, so the DNA content is still 2C?
Correct. The ploidy is halved.
You know, I have to pause on that point. It seems so counterintuitive, especially if you're used to thinking about mitosis. The cell spends enormous effort breaking bonds, but here, it actively has to prevent the sister chromatids from separating at the centromere.
It does.
Why take that risk? What is the molecular guardian? What is the thing that keeps them glued together?
That is a fantastic question, and it gets right to the heart of the regulatory mastery of meiosis. The protein you're looking for is called chugoshin,
Shugoshin.
which, fittingly enough, is Japanese for guardian spirit.
That's perfect.
It is. So in a normal mitotic anaphase, an enzyme called separase comes in, and it cleaves the cohesin proteins that are holding the sister chromatids together.
The molecular glue.
The molecular glue.
Yeah.
During meiosis the est, Shugoshin steps in, and it protects those cohesins, but specifically at the centromere region. It shields them from separase. This molecular protection ensures the sisters stay attached, allowing only the homologs to separate first.
So without Shugoshin.
Without Shugoshin, the sister chromatids would separate prematurely in anaphase the first, and the entire meiotic process would collapse into catastrophic aneuploidy. It's an absolutely vital mechanism.
That's incredible. It really underscores that meiosis isn't just one separation event. It's a precisely timed two-step separation. The first division is separation of the pair, and the second is the separation of the twins.
That's a perfect way to put it.
Yeah.
So after meiosis the first, telophase I finishes the job, the cell splits into two haploid cells. If there's an interphase 2 between the two divisions, it is very brief, and it never involves DNA replication.
Right, because the chromosomes are already duplicated from that initial S phase.
They're ready to go.
So meiosis II at the separation division is essentially just performing that final mitotic-like separation, but it's doing it in two cells that are already haploid.
Correct. The stages prophase 2, metaphase 2, anaphase 2, and telophase 2, they look just like their mitotic counterparts, but with N chromosomes instead of 2N.
So in metaphase 2, the individual chromosomes, each with two chromatids, they finally align single file at the center.
They do. And now, finally, the kinetochores of the sister chromatids face opposite poles, just like in mitosis.
Setting the stage for anaphase II.
And in anaphase 2, those remaining cohesins at the centromere are broken, the sister chromatids finally separate, and they move to opposite poles, officially becoming individual, unduplicated chromosomes.
And the final outcome?
The final outcome is the triumphant production of 4 genetically unique daughter cells. Each one is haploid, 1N, and has a DNA content of 1C, and each contains a random assortment of chromosomes derived from both parents, which have been further diversified by all those crossing over events.
Let's just quickly track that DNA content change using the C value again, just because it really highlights the reduction.
Good idea.
We start the cell cycle in G1 at 2N, 2C. After DNA replication, we're at 2N, 4C. Meiosis mypo cuts the ploidy in half, leaving us with two cells that are 1N, 2C.
Correct.
And then meiosis II cuts the DNA content in half, resulting in the 4 final gametes, each at 1N, 1C. If that process wasn't crystal clear before, that 2C to 1C halving in meiosis II is its entire purpose.
That's its job.
We've spent a lot of time admiring the mastery of this process, but like in any complex physical performance, mistakes can happen.
And they do.
And when they happen in meiosis, the consequences for the resulting offspring can be catastrophic.
Absolutely. The occasional failures in chromosomal partitioning are known as nondisjunction.
Nondisjunction.
It leads to gametes that are carrying either an extra chromosome or are missing a chromosome. This results in an abnormal chromosome number, a condition called aneuploidy, in the zygote after fertilization.
And this can happen in either division.
Yes. Nondisjunction can occur during meiosis I if the homologous chromosomes fail to separate correctly, or it can happen during meiosis II if the sister chromatids fail to separate. The results are defined by the ultimate number of chromosomes.
Okay, so what are the terms for that?
If the resulting zygote gets an extra chromosome, it's called trisomy, so 2N plus 1. If it's missing a chromosome, it's called monosomy, 2N1.
And monosomy is usually worse, isn't it?
Much worse. Monosomy is often lethal very early in development because the dosage imbalance of losing an entire chromosome's worth of genes is just too severe for the embryo to handle.
You know, I think we need a quick analogy here because the difference between aneuploidy and a related term, polyploidy, can be a little confusing.
That's a great idea. Okay, think of your genome as a large, specific bookshelf, say 23 shelves, and each shelf holds a matched pair of books. Those are the homologous chromosomes.
Okay.
Aneuploidy is like going to just one shelf, say shelf 21, either pulling out one book or adding an extra third copy of that specific book. That functional imbalance is highly disruptive because the dosage is wrong for just one set of genes. The whole library is out of whack.
Right.
Polyploidy, on the other hand, means the organism contains complete multiple sets, maybe 3N triploid or 4N tetraploid. This is like taking that entire 23-shelf bookshelf and flawlessly duplicating it to have 2 or 3 complete matched bookshelves.
So the ratios are still balanced.
The ratios of all the genes are still perfectly balanced. While it's not normal, the relative functional imbalance is often much less severe. And this is why polyploidy is very common and often benign in plants. But aneuploidy is usually lethal or associated with severe syndromes in animals.
Speaking of which, the most common trisomy we see in live human births is Down syndrome, or trisomy 21. It happens in about 1 in 700 births.
That's right. It's an extra copy of chromosome 21. And critically, most cases of Down syndrome are caused by nondisjunction during female meiosis the first.
And this failure is strongly correlated with maternal age.
Very strongly. The frequency climbs dramatically, especially after age 40. The working theory is that those oogocytes, which have been arrested in prophysis for decades, experience an age-related degradation in the machinery, like the cohesins or the spindle apparatus, that just makes separation mistakes more likely over time.
And the symptoms we see, the physical and cognitive deficits, are a direct consequence of that single molecular mistake.
It all comes down to gene dosage. It's linked to having 3 copies instead of 2 of genes located in a specific area called the Down syndrome critical region, or DSCR, on chromosome 21.
This circles us right back to that insurance policy of the diploid state. The diploid state works because 2 is the perfect number. It gives you just the right amount of expression.
Exactly.
But when you introduce a 3rd copy, it means certain proteins, like a kinase called DYRK or a cell surface protein called BISCAM, are being overexpressed by 150%. This massive dosage imbalance just cascades through cellular pathways and disrupts the delicate balance required for normal development.
And while trisomies of the autosomes that result in live birth are pretty rare, aneuploidies involving the sex chromosomes are much more common.
Why is that?
It's largely because the Y chromosome is very small and doesn't have many genes, and females have a natural mechanism called X inactivation to functionally silence one of their 2 X chromosomes anyway. So the system is already built to handle some dosage variation there.
Which leads to conditions like
Klinefelter syndrome, which is XXY. It results in sterile males who often have some female characteristics. And Turner syndrome, which is monosomy X, results in sterile females who are often of short stature. But the fact that Turner syndrome can be treated with estrogen replacement therapy really underscores the direct, measurable impact of these meiotic outcomes on the body.
Okay, let's shift from the mistakes of meiosis to the mastery of it, specifically in how functional gametes are created. This is gametegenesis. And the differences between male and female production here are, well, they're staggering.
They really are, and they reflect a deep functional priority.
So in spermatogenesis, the male production, the process is highly symmetrical.
Very symmetrical. A diploid spermatocyte goes through meiosis vanicin 2, and the result is 4 equal-sized haploid spermatids.
And then those have to mature.
Right. They undergo a differentiation process where they shed most of their cytoplasm, develop the flagellum they need for motility, and become these high-volume, lean, functional sperm cells.
But eugenesis, egg formation, is the complete opposite. It's drastically asymmetrical.
Totally asymmetrical. The cell divides its cytoplasm unequally during both meiosis the first and meiosis the second. And the result is only 1 large functional egg cell.
And the other 3?
The other 3 are just small, non-functional polar bodies that simply degenerate. They're basically little packages of discarded chromosomes.
And this asymmetry is a clear case of structure supporting function, isn't it?
It's a perfect example. The female reproductive strategy prioritizes resource concentration. By funneling virtually all of the stored nutrients, all the mitochondria, all the regulatory components into that one single massive egg cell, the organism maximizes the developmental resources available to sustain the embryo until it can implant or hatch. The human egg's volume is just immense compared to a sperm.
The timing of oogenesis is also highly specialized. We can see this really well in model organisms like frogs, which have taught us so much about cell cycle control.
Frog oocytes undergo these prolonged periods of growth while they're arrested in prophase I. That's when they accumulate all those essential resources and protective coatings.
And some landmark experiments in the 1970s dramatically illustrated how this maturation is regulated.
They were brilliant experiments.
Researchers found that if they took cytoplasm from a mature dividing egg and injected it into an immature arrested oocyte, the immature cell would immediately resume meiosis.
even without any of the normal hormonal cues. This discovery led directly to the identification of maturation promoting factor, or MPF.
And what's so fascinating is that MPF turned out to be chemically identical to the mitosis promoting factor used in standard cell division.
It's the same machine, a protein complex made of a CDK, a cyclin-dependent kinase, and a cyclin subunit. In this case, the hormone progesterone acts as the external signal. It triggers the production of the necessary components to activate MPF, and that activation allows the cell to finally blast past that prophase I arrest and complete meiosis the first.
But the regulation isn't over yet. In most vertebrate eggs, including ours and frogs, the cell immediately proceeds to a second arrest point.
A second roadblock, this time in metaphase 2.
And what holds it there?
This second arrest is maintained by something called the cytostatic factor, or CSF. And one of the critical proteins in CSF is a molecule called EME2.
EME2.
EME2 acts as a molecular brake. Specifically, it inhibits the anaphase promoting complex, or APC.
Which is the complex needed to get out of metaphase.
Exactly. Since APC activity is absolutely mandatory for the cell to transition from metaphase into anaphase, inhibiting it halts the entire division process right there at metaphase 2.
So the egg cell just sits there, poised at the very edge of final division, potentially for hours or even longer, just waiting for its final signal.
Waiting.
So what's the trigger? What is the signal that finally frees the ATC and lets the cell complete its meiotic journey?
Fertilization.
The entry of the sperm.
So that's the switch. When the sperm enters the egg, it triggers a massive cascade involving a huge release of calcium ions inside the cell. This calcium surge activates specific kinases, which in turn phosphorylate EME2.
And that's the kill signal for ME2.
It's the kill signal. Once EME2 is phosphorylated, it's instantly targeted for destruction. With that break removed, the APC is finally active, allowing the cell to transition into anaphase II, complete meiosis, and integrate the sperm genome into the newly formed zygote. It is an elegant, explosive, and absolutely final regulatory checkpoint.
Okay, so we've established that meiosis is essential for balancing the chromosome count, but its role in generating genetic diversity is, it's arguably its greatest evolutionary gift.
No question.
And just to remind everyone that diversity comes from two major physical events: random assortment back in metaphase 1 and the crossing over in prophase I. But to truly appreciate the physical consequences of this chromosomal ballet, we really have to look back to the abstract work of Gregor Mendel in 1865. He did this long before anyone had even seen a chromosome.
It's incredible. His genius was that he derived the laws of inheritance just by counting peas in his garden.
So he started simply crossing two true breeding parental strains, the pea generation, for example, a purple flower with a white flower.
Right. And when he crossed those, the F1 generation were all hybrids, and they all displayed only the dominant trait. They were all purple. This result really baffled a lot of scientists at the time who believed in a blending theory of inheritance.
But the real insight, the real genius, came when he let those F1 hybrids self-fertilize.
That's right, because in the F2 generation, the recessive white trait miraculously reappeared, and it reappeared consistently in a 3-to-1 ratio of dominant to recessive phenotypes. This was proof that the trait hadn't vanished. It had just been hidden or discrete.
And Mendel correctly deduced that these traits are determined by factors, which we now call genes, and that the alternative versions of those factors are what we call alleles.
And the physical address on the chromosome where a gene resides is its locus.
All of this led directly to his first major principle, the law of segregation.
The law of segregation states that the two alleles for a trait remain distinct entities, and they separate into different gametes during meiosis. So a parent who is heterozygous, PP, will produce gametes carrying a big P and gametes carrying a little p in equal measure.
And this physical separation of the alleles during gamete formation is precisely what allows that recessive trait to pop back up in the F2 generation.
It's the only way to explain the math. Mendel then extended this to two-factor crosses, testing plants that differed in two distinct characteristics, like seed color and seed shape.
And based on the ratios he saw in the F2 generation there, he formulated his second law.
The law of independent assortment, which says that the alleles of each gene separate independently of the alleles of other genes.
In other words, whether you inherit the allele for green seeds doesn't influence whether you inherit the allele for wrinkled seeds.
Right. But there's a crucial limit to this law that Mendel couldn't possibly have known. It only holds true for genes located on different chromosomes, or genes that are located very, very far apart on the same chromosome.
And the ultimate physical explanation for Mendel's abstract laws came decades later, in the early 1900s, with the development of the chromosomal theory of inheritance.
Championed by scientists like Boveri and Sutton, it provided the physical mechanism for everything Mendel had observed just by counting.
The theory states that cells have two sets of homologous chromosomes, that these chromosomes are genetically continuous, and that the homologous chromosomes are functionally equivalent. But
Most importantly, it identified the physical events in meiosis as the direct cause and effect link.
so the synapsis and the segregation of homologous chromosomes during meiosis the first.
That provides the physical basis for the law of segregation. The two alleles are on the two homologous chromosomes, and meiosis I separates them.
And the random orientation of different homologous pairs at metaphase Y.
That provides the physical basis for the law of independent assortment.
The randomness we talked about earlier.
Exactly. You can visualize it perfectly. If you track a gene for seed color on chromosome 1 and a gene for seed shape on chromosome 7, meiosis I forces those two chromosome pairs to align independently at the metaphase plate. There are two equally likely orientations, and that physical randomness guarantees that all four possible combinations of alleles will be produced in the gametes with equal probability. The movement of the chromosomes is the inheritance.
Which brings us perfectly to the limit of that independent assortment. Linkage.
Linkage. As soon as you discover genes that are located on the same chromosome, they tend to be inherited together. If genes D and E are both on chromosome 5, they are said to be linked, and the alleles for D and E will stick together most of the time.
And this was the groundbreaking discovery of Thomas Hunt Morgan and his team using the fruit fly, Drosophila.
A perfect model organism for genetics. They found that mutant genes did not assort independently. Instead, they segregated as these linkage groups.
And here's the profound finding, right? The number of linkage groups they identified was 4.
4, which perfectly matched the haploid chromosome number of the fruit fly. This was definitive proof that the chromosome is the physical entity that carries the linkage group.
But Morgan also noticed something critical. The linkage wasn't always complete. Occasionally, the linked traits would appear in the offspring in these non-parental combinations.
Which suggested that the homologous chromosomes were physically exchanging segments during meiosis. He called this mechanism crossing over, or recombination.
Which is that breakage and fusion event we detailed way back in Prof S1.
Exactly. So if you start with linked parental alleles, say big A and big B on one chromosome, and little a and little b on the other, a single crossover event between them ensures that when meiosis II is complete, you will end up with 4 gametes.
2 will be the original parental type.
Right, you'll have 2 parental gametes, AB and AB, but you'll also have 2 recombinant gametes, AB and AB. The creation of those recombinant gametes is the physical result of the chiasma.
And the observation that the frequency of this recombination varied for different pairs of genes. That sparked the genius of Alfred Sturtevant, who was an undergraduate in Morgan's lab at the time.
An undergraduate. His insight was simple, but absolutely revolutionary. He realized that the farther apart 2 genes are on a chromosome, the more physical space there is between them.
So a crossover event is just more likely to occur between them.
It's more likely to happen, thus separating the linked alleles. This gave rise to the entire science of genetic mapping, using the recombinant frequency to determine the sequential order and the spacing of genes all along a chromosome.
And we quantify this distance. A 1% recombination frequency is defined as 1 map unit.
Also known as a centimorgan, in honor of Thomas Hunt Morgan. So if you run an experiment and you observe that 15% of your offspring have a recombinant genotype, meaning the alleles didn't stay linked, you can conclude the genes are 15 map units apart.
It's incredible. This method, developed entirely by tracking how often traits stick together, allowed scientists to build these detailed linear maps of chromosomes.
And today, even with affordable direct DNA sequencing, what we call physical mapping, the results derived from those old recombination frequencies align remarkably well with the physical reality of the chromosome. The structure of the chromosome is linear, and the frequency of exchange is a remarkably accurate proxy for physical distance.
It's important to remember, though, that while this classic sexual process relies on meiosis, the fundamental molecular process of recombination, the breaking and rejoining of DNA, is widespread. It happens even in organisms that are primarily haploid and reproduce asexually.
Right. Bacteria and viruses, they lack the full meiotic cycle, but they still have very effective ways to mix and match their DNA.
So let's start with viruses. How do phages, which are really just DNA in a protein coat, manage to recombine their genetic material?
In bacteriophages, recombination happens when a single bacterial cell is simultaneously infected by two related phages that has slightly different genotypes.
A co-infection.
A co-infection. Once the phage DNA is inside the host cell and starts replicating, the homologous DNA molecules can physically align with each other. Crossing over occurs, and the result is recombinant phage particles that carry a mix of the two original genotypes. This just proves that recombination is an ancient, fundamental molecular process.
For bacteria, which are incredibly adept at swapping genetic information, they have three primary non-sexual mechanisms to do this. The first one is transformation.
Transformation is simply the uptake of free, naked DNA fragments from the environment by a bacterial cell.
And once it's inside,
that exogenous DNA has to be integrated into the host genome via homologous recombination, requiring two crossover events for it to survive and be expressed. This is a natural mechanism bacteria use to acquire new traits, like antibiotic resistance genes, if they happen to encounter the right DNA in their environment.
The second mechanism is transduction.
Transduction is DNA transfer that's mediated by a bacteriophage. Sometimes, when a new phage is being assembled, it accidentally packages fragments of the host bacterial DNA instead of its own viral DNA.
A packaging error.
A packaging error. And when this faulty phage then infects a new recipient bacterium, it injects that host DNA fragment. And again, that fragment can then be integrated into the new recipient's genome via recombination.
And scientists can leverage this, right?
They do. They use transducing phages, like phage P1, for what's called co-transductional mapping. The logic is simple. If two genetic markers are very close together on the bacterial chromosome, they are much more likely to be packaged together and travel together in the same phage fragment. By measuring how frequently two markers are co-transduced, scientists can infer their proximity, much like using recombination frequency in eukaryotes.
And the third major mechanism is conjugation. This is the closest thing bacteria have to sex.
It is. It requires direct physical contact between a donor bacterium and a recipient. And this contact is established by a thin cytoplasmic mating bridge formed by specialized structures called sex pili.
And the ability to be a donor to build these pili relies on a specific piece of mobile DNA, right?
Correct. That ability is conferred by the F factor, which stands for fertility. It's a circular DNA plasmid. A cell that carries this plasmid is called F+, and it's the donor.
So what does it do?
It transfers a copy of this plasmid across the mating bridge to an F- recipient. And because the plasmid is essentially an independent, infectious piece of DNA, the F+ cell converts the recipient into a new F+ cell.
So maleness, in this case, is infectious.
It's infectious,
right,
which is a critical factor in epidemiology because it explains why resistance can spread so quickly.
Right, because similar plasmids, called R factors, are the ones that carry those antibiotic resistance genes.
Precisely. And R factors often contain transposable elements that allow them to integrate new resistance genes quickly and efficiently. It's a huge part of the global antibiotic resistance crisis.
Now, the transfer becomes even more significant when that F factor plasmid integrates itself into the main bacterial chromosome.
Yes. This converts the F+ donor cell into what's known as an HFR cell. It stands for high frequency of recombination.
And when an HFR cell tries to conjugate, it doesn't just send the plasmid. It tries to transfer the entire bacterial chromosome, starting at the F factor's integrated origin of transfer.
It does. But since the mating bridge is fragile and usually breaks spontaneously before the full transfer is complete,
only a segment of the chromosome gets transferred.
Exactly. And this allows scientists to map the circular bacterial chromosome linearly. The genes that are closest to the origin of transfer are the most likely to be successfully transmitted to the recipient before the bridge breaks. By timing the duration of mating and measuring which genes get transferred, you can construct a reliable map of the gene order and spacing.
And once that linear fragment is in the recipient cell, it has to recombine with the recipient's genome to be successfully integrated.
It does. Recombination is the final step.
Okay, we've established that genetic recombination happens everywhere, from the human oocyte to the HFR bacteria. But what is the definitive molecular proof that this is a physical breakage and exchange and not just some kind of new DNA synthesis?
The foundational evidence for that came from two classic experiments back in the 1960s. First, Messelson and Weigel co-infected bacteria with two strains of phages. One strain was labeled with a heavy isotope of nitrogen, 15N, and the other with a normal light nitrogen, 14N.
Okay, so heavy DNA and light DNA.
Exactly. After recombination was allowed to occur, they analyzed the resulting phage DNA, and they found that the recombinant DNA molecules, the new combinations, contain a mixture of both the heavy and the light isotopes,
Meaning pieces have been swapped.
meaning the genetic material had been physically broken and rejoined. And since they designed the experiment to prevent new DNA synthesis, this definitively ruled out synthesis as a primary mechanism. It proved it was breakage and exchange.
And then J. Herbert Taylor showed the same physical mechanism was at work in eukaryotes.
He did. He used radioactive thymidine to label DNA strands before meiosis. And after the division, he saw that the resulting chromatids contained a patchwork, a mixture of labeled and unlabeled segments. This pattern was only possible if there was a physical reciprocal exchange between the homologous partners.
So now we understand what happens. Let's look just a little closer at how the cell achieves this high-fidelity exchange, because it sounds dangerously close to just breaking the genome.
It is a high-risk operation, no question.
Yeah.
And that's why the molecular machinery is so critical.
Yeah.
As we discussed, the synaptonemal complex, the SEC, acts as that molecular vice grip. It ensures the homologous chromosomes are perfectly aligned along their entire length during pulchitine. Without that perfect alignment, any exchange would be catastrophic.
And the cell doesn't invent a new process for this.
It doesn't. What's fascinating is that meiotic recombination relies almost entirely on the same molecular pathways the cell uses for high-fidelity DNA double-stranded break repair.
So, wait, meiotic recombination is basically just a controlled, intentional DNA repair process.
That's the elegant conclusion. The cell intentionally creates a controlled break, and then it uses the homologous chromosome as the template to repair that break. But in doing so, it swaps the genetic information.
And how does it find the right template?
The initial break triggers a process called homology searching. The free strand of DNA actively seeks out and checks for complementary sequences. This searching is mediated by two absolutely critical proteins, RBK in prokaryotes and RED51 in eukaryotes.
So RED51 is the search party?
RED51 is the search party. These proteins coat the single-stranded DNA and actively facilitate the invasion into the homologous double helix to check for a match. And we can see this happen. When scientists stain for RED51, they see these little clusters or foci right on the condensed chromosomes during early pagitine. It's direct visual evidence that this repair machinery is active precisely when and where crossing over occurs.
And this whole process is highly regulated.
Extremely. We know recombination isn't uniform across the genome. There are recombination hotspots with unusually high activity. And conversely, too much recombination is detrimental to genome stability.
Which we see in human diseases.
Exactly. A lack of specific DNA helicases associated with a condition called Fanconi's anemia leads to abnormally high rates of recombination and genomic instability.
So the final summary is a real masterpiece of structure and function. The synaptonemal complex facilitates the alignment and controls the breakage and resealing process, creating the chiasmata. And those chiasmata, the visible result of the exchange, are not just genetically vital for creating diversity, they are structurally vital for holding the homologous chromosomes together on the metaphase I plate, ensuring they separate cleanly and correctly.
That's the whole story in a nutshell.
Okay, let's recap the 4 major takeaways from this deep dive.
All right. First, meiosis is essential for generating haploid gametes. This balances the chromosome number over generations and maintains the stability of that all-important diploid state. Second, the massive evolutionary power of sex stems from genetic variation, which is generated physically by 2 key events: the random orientation of homologous chromosomes at metaphase of first, and the physical crossing over that happens during prophase of first.
Third takeaway.
Third, Mendel's abstract laws of inheritance, segregation, and independent assortment find their physical explanation entirely in the movement and segregation of chromosomes during meiosis the first and 2.
And finally,
Recombination is a fundamental, tightly regulated molecular process. It borrows its machinery from high-fidelity DNA repair pathways, and it operates across all forms of life, from HSR bacteria to the highly complex human oocyte.
we've seen today that successful meiosis is absolutely key for human health, and that failure leads to devastating conditions like Down syndrome because of that catastrophic gene dosage imbalance. But let's close with a final provocative thought. Building on that structural role of the chiasmata, if that physical connection provided by crossing over the chiasma is absolutely crucial for holding homologous chromosomes together at metaphase I to ensure they don't separate incorrectly, what happens in a species that completely skips this step?
Okay.
For instance, Male fruit flies are famously known to entirely lack meiotic recombination. They never cross over.
Right.
So, without the chiasmata acting as the physical glue, what molecular or structural solutions must they have evolved to ensure their homologous chromosomes still manage to find their way to the metaphase plate and segregate correctly into the daughter cells? It forces you to connect that essential structural function, holding the partners together, to survival itself. And it proves that somehow the cell always finds a way.
A fascinating question to ponder.
Thank you for joining us on this deep dive into the mechanics of inheritance and variation. We hope this knowledge provides you with a robust framework for understanding the very core of cellular life.